Hello, good afternoon. We have a relatively short lesson today. We just have to wrap up lecture four, um, and I will tell you about the lab. The rest of lecture four, like we have the PowerPoint slides, but I think it's better if we just, take a note together, and then, you can go through the slides on your own um, later on. Okay, so please take out a piece of paper. Uh, we are gonna be doing a flow chart, and put your paper sideways like this, and then start in the middle, and then we're gonna do a flow chart. Okay, so it's gonna go all the way like this. All right, so just so if you're planning ahead for the note, that's what we're gonna be doing. Actually, you know what? I can show you the note because I took it with my other class, so you have an idea of how big this is going to be. Okay, so that's what we're doing. Right? We're doing this whole chart. Right? Starting with transport, and then we're going to go all over the place like that. Okay. All right. Now maybe we take a moment and recap what we did last time first, right? Last time we were talking about the membrane protein functions, and then we moved on to talk about the importance of ATP, which helps various cellular work in the body, such as movement. We call those mechanical work. There are transport work, moving things across the cell membrane sometimes requires ATP, and there are also chemical work for doing chemical reactions. And then we talk about tonicity, right? We looked at what happened to a cell when it's like in an isotonic, hypotonic, and hypertonic solution. So today we're just gonna basically look at the different types of transport that exist. Specifically, how do things cross the cell membrane to enter the cell or to leave the cell? So let's start with the word transport here. Transport across the cell membrane. And we're going to put a box around it. So there are two types of transport in general, two major categories. Let's start with the easier one first. It is called passive transport. Passive transport. Everything I'm writing down for you um, is also in the PowerPoint slide. Uh, I think going through the slides is not as effective as drawing it out, writing it out together, right? For some things, anyways. In fact, for lecture five, we're going to be uh, writing notes for the whole class because it's just like such a difficult concept. We do have the PowerPoint slides, but it's for you to go through on your own later. Okay, we'll be taking notes primarily. Anyways, back to this thing. Passive transport basically has the following features. Okay, um, it goes from high concentration. Okay, remember the square bracket means concentration. High concentration to low concentration. Okay, we say substances will move down is concentration gradient. Down the concentration gradient. If you forgot what a concentration gradient is, it's basically a difference in concentration between two areas. So it's going from high to low. It's like a ball rolling downhill. Also, like a ball rolling downhill, there's no energy required. You give it a bit of a nudge, and then gravity does the rest, right? So in the same sense here, when substances move from high to low concentration, no ATP needed. Okay? It just happens spontaneously. So there are different types of uh, passive transport. For example, we have osmosis. Osmosis is basically the diffusion of water. Right? So example would just be water. Only water can do osmosis. Okay, like I mean, other things we don't call it osmosis, right? It's only when water does it, then we call it osmosis. So that's passive. All those hypertonic, isotonic stuff that we talked about, it just happens on its own. Right? No energy is required. And then we have simple diffusion. Simple diffusion. Right? So simple diffusion basically is best for small molecules. Right? It's good for hydrophobic or nonpolar molecules. Okay? If it's hydrophobic and nonpolar, they're able to cross the phospholipid bilayer easily. Okay, because remember, the phospholipid bilayer um, has a thick layer of hydrophobic tails sandwiched in between. And so anything that's hydrophobic will have no problem crossing that. So example of hydrophobic molecules that can cross include things like steroid hormone, nonpolar molecule would be things like carbon dioxide and oxygen. Okay. So if it's small or if it's hydrophobic, nonpolar, they can go in or out on their own. That's called simple diffusion. Now, sometimes it might be too big. The molecule might be too big to cross the membrane on its own, or sometimes it might be charged. And so we need a different kind of diffusion. That's why this is like simple diffusion. And then we have something called facilitated. Facilitated diffusion. Okay. Let me just give you the definition. Facilitated diffusion involves moving substances down, we can say across the membrane, across the cell membrane. Now remember, we are talking about passive transport still, so this is going down the concentration gradient. I'm not going to write it, okay, but just keep in mind that it is. So moving substances across the cell membrane down their concentration gradient, but with the help of membrane proteins. Okay. So these things are like usually too big to cross on their own, or they are charged, so they can pass through the hydrophobic tail layer of the membrane, and so they need a little bit of help. And there are two kinds of facilitated diffusion. The first kind is called carrier proteins. So in carrier proteins, the molecule binds to the carrier protein and causes the protein to change shape. In doing so, the molecule is transported across the membrane. Now, perhaps it's easier to show you what it looks like. I'll give you a moment to copy it down first, and then I'll draw you a picture to explain what it looks like. All right. So we will draw a little bit of the phospholipid. Okay. Not as big as what we did before. Okay, just like 6 of these things maybe. And then another few here. Okay, so here is the carrier protein. Okay, maybe just watch first. I'll give you some time to copy after. So it opens up on one side, and let's say I have a molecule here. Okay, so remember, it's still going from high concentration to low concentration, all right? So let's say you have a lot of these guys outside. Okay, this is outside of the cell, and this is inside. And you know, we have some inside, just not a lot. Right, so high concentration for the triangle stuff on the outside and low concentration on the inside. And what's going to happen is it will bind to the receptor protein, it will grab onto it, and then it will cause the protein to change shape. So instead of opening outside, now it's going to open inside, right? And then in doing so, oops, wrong color. In doing so, the material will now be transported across the cell membrane. Okay? It's like those revolving doors, like at some malls, right? Like you go in one side and then you push the door and then it rotates and then you end up inside the mall, right? So you in this analogy, you are the molecule and the revolving door is the carrier protein. So you bind to the door, okay? You cause the door to shift to rotate, and then before you know it, you are inside. Does that make sense? Does anybody have any question about what I described here? Do you need me to clarify or anything at all? Okay. So that's one example. And you know, molecules that would use this would be something that's big. So like glucose. Okay. This is ideal for big molecules. Okay. Big molecules. They are too big to cross on their own. They need a bit of help. All right, so copy what you need to. And then I don't know, give me a sign when you're ready, okay? Like tell me you're ready, give me a thumbs up or something. Because I know sometimes people can't copy everything um, if I'm going too fast, I suppose. We good? Can I continue? Yeah? Okay, thank you. Next, we have another one, which is called the channel protein. Okay, channel protein. Just to remind you, the carrier protein and the channel protein, they are both examples of facilitated diffusion. Okay, the difference between facilitated and simple is that you need to have a membrane protein to help you in facilitated diffusion. Okay, so for channel protein, what happens is that uh, the channel protein provides a channel. Okay, channel is another way to say tunnel. Okay, provide channel or tunnel for charged molecules to pass through. Okay, so usually, if it's charged, that means it's like, you know, not gonna interact well with hydrophobic stuff. Right? And as you can see, the phospholipid bilayer, there's a thick hydrophobic tail section in between. So if it's charged molecule, it's not going to be able to go through. Yeah? So it needs to have a tunnel. So example would be like, calcium or something. Let's draw a picture for this as well. This one's easy to draw. So there it is. We have an integral protein. That means it goes all the way through, and there's a tunnel in the middle. Tunnel in the middle. All right, so let's say you have a lot of these guys. Let's make it, there we go. Lots of them. You know what? Actually, I'm not gonna have a lot of them. I'm just gonna have a few because I don't want you to get the misconception that it's always going out into the cell. Like the direction, it's not always in. It could be out as well. Okay, it depends on which side has higher concentration, right? And it's always gonna go from high to low. With passive transport. So these things are going to move through the channel and then it's going to end up outside. Okay? These are like calcium ions, okay? If it's charged, it can cross on its own. If it's too big, it can cross on its own. So we need to use facilitated diffusion, okay? Either through carrier protein that literally carries you through or the channel protein, which is just going to be there. There's a tunnel. You walk through it. You're on the other side. That's all for the passive transport, okay? These are all the different types of passive transport. Okay, we just have to do the active transport. And then we'll be done. Again, like because of my other class, they lost some class times, right? So I can't go too fast with you guys. I have to try to keep pace. That's why we're, cutting the class a little bit shorter. So here, the other kind of transport is active transport. Active transport. You guys will also have a review class for the test, which is great because of the extra time that we have. So for active transport, just the contrast with this, instead of going from high to low concentration, it is going from low to high concentration. Low to high. So we are going against the concentration gradient. And it requires ATP. ATP needed.
Passive was high to
I'm sorry, what is it?
passive was high to low, no? Passive
Yeah,
transport.
passive is high to low, that's right.
Yeah.
And active would be low to high. Is that what I wrote before? Yeah, look, high to low for passive, low to high for active. It's like pushing a boulder uphill. That's gonna take some energy, right? As opposed to rolling downhill, no energy required. All right. If you're planning for space, right, active, so we're gonna have one here. We're gonna have another one here, which separates into 1 and 2, and then this guy's gonna separate into 3, okay? Just so you are planning ahead. You don't run out of space. Okay, so here we go. Over here, we have an example called the protein pump. Okay. So I will give you 2 examples of protein pump. Okay. I mean, the description is pretty straightforward. The description is basically active transport. The protein pump is a membrane protein. Okay, I'll write down for you. An integral protein that uses ATP to move things against their concentration gradient. I'll give you 2 examples of this. Example 1, it is a, a proton pump. Okay. So in the stomach, that's the stomach, okay? In the stomach, do you know we have acid in the stomach? I hope you do. There's some stomach acid here. And the stomach acid needs to have a lot of H plus in it. Okay, so along the walls of your stomach, the cells would have a proton pump, and they would continuously put proton into the acid. Okay, the more proton you have, the more H plus you have, the more acidic it's going to be. So that's an example of active transport because you already have a lot of H plus in the acid, but you want to make it even more acidic. So you keep on pumping more into it. People who have heartburn a lot, they might have to take PPI, which is proton pump inhibitor. Another example of this is something called the sodium potassium pump. Okay. We're going to learn more about this next semester, but for now, cell wants to, I guess cells, right? Cells want to keep K plus while removing Na plus. Okay, exactly why they want to do that, I'll tell you next semester. But let's see what happens. Okay, so here is our phospholipid bilayer, and we're gonna draw a sodium-potassium pump here. Okay, so I'm gonna put like 3 square slots, and then 2 circle slots. Okay, I know it looks really weird. Okay, but let's see if this makes sense. Okay, so we're gonna have 2 circles here that fits into the circle slot, and then we're gonna have 3 squares that fits into the square slots of the pump. Okay, the purple thing is the sodium potassium pump. And what's going to be happening is we will be pumping these blue things out, and we'll be bringing the circle in. Okay, so those circles are actually, believe it or not, they are actually potassium. Okay, so these are potassium. Okay, this is inside, and this is outside of the cell. And what happens is as the pump pumps potassium in, it'll pump 2 at a time, okay? Over time, you will have high potassium inside and low potassium outside, right? Which totally makes sense because you're bringing all of them in, right? And so if you want to continue to do this, you are actually moving potassium against its concentration gradient, right? Going from low to high. And at the same time, we will have a bunch of sodium here. And we actually would have more on the outside. Right, so for every 2 potassium that you bring in, 3 sodium are going to go out. Okay, 3 sodium and 2 potassium. So the story is the same for the sodium. If you keep on pumping them out, you will have low sodium on the inside and you will have high sodium on the outside. So this requires ATP. Okay? Because you are going against the concentration gradient for both of these ions, right? For the sodium as well as for the potassium. So these are examples of protein pump. Okay? They are membrane proteins, often integral proteins, and they're going to be pumping things across the membrane, against their concentration gradient. Any questions? Everybody happy? All right. Next, we have bulk transport. Bulk transport. Gonna go all the way over here. Bulk transport. As the name suggests, bulk transport is to move large amount of substances across the cell membrane. And you can be bringing things in or sending things out. Okay, so if we send things out, it's called exocytosis. Exocytosis. This is to send things like large amount of it, okay? Send things out of the cell. So when would you want to do this? Well, remember that vesicle that we created from the rough ER? It's going to go to the Golgi. It's going to get repackaged. And once you repackage it, there are different places it can go, right? And one of the places it can go is to be sent out of the cell. And that would be an example of exocytosis. Okay, let me try my best here to draw it for you. Okay, so this is the cell membrane. Okay, and so you have a vesicle, and inside the vesicle is going to be a bunch of proteins or whatever, right? Doesn't have to be proteins, it could be other stuff as well. So this is going to move towards the cell membrane. And by the way, this is inside, this is outside, and this is cell membrane, okay? So the vesicle will move towards the cell membrane. It's guided by those microtubules and whatnot, and then it will touch the cell membrane. And after it touches the cell membrane, it actually will fuse with the cell membrane. It will fuse with the cell membrane. I'm just showing you like in still picture what happens one step after the other. And then eventually it will be like completely fused with the membrane while releasing all of these things into the surrounding. Okay, so vesicle is going to go and then opens up, send the stuff out. This is called exocytosis. Are we okay? I know I ask you that every 5 minutes, but seriously,
Yes.
thank you. Uh, so it's always good to hear, even one or two students. All right, next we have the endocytosis. Endocytosis, which is as the name implies, um, endo is to bring it in, okay? Exo is to exit, right? So instead of sending things out of the cell, here we bring things. Again, because it's under bulk transport, when I say bring things, I'm talking about bringing large amount of things. Bring things into the cell. Okay, so pretty much is we're going to draw it in reverse. And let's say we have these. Let's bring some blue stuff in, okay? There's some blue stuff here that you want to bring in. And what's going to happen is that the cell will actually kind of create a groove, and then the blue stuff will sink into it, and eventually, you will close off the area like this, and then the vesicle will be internalized. Okay, again, you really have to kind of use your imagination here. I mean, like if you Google exocytosis or endocytosis, I'm sure there are lots of videos on this, but hopefully You can see it, right? Oh, sorry, I see a message. It's really hard to remember all these things. Yes, I know. I am sorry. Again, I didn't make up these things, right? Like you go anywhere to do the pre-health program, they will teach you the same thing, okay? Because it's standardized, the curriculum. But like I said, take advantage of the fact that you are doing this online. So have a bit of a reference sheet next to you to just take a quick peek if you need to. Okay, I totally understand. There's a lot of stuff here, and some of it is a little bit abstract. Um, Trust me, I am with you there. Okay, I don't know. Hopefully, that's a little bit of comfort to you. If not, I'm sorry. Okay, so that's endocytosis. Now, we have this is it. Okay, this is the last one. I can hear you guys screaming at me through the screen. There are three different types of endocytosis. Okay, three different types, and that's it. Okay, three different types. It depends on what and how you are bringing it in. Is this like a different color? I think it's a different color, but oh well, whatever. It doesn't matter. So let's start with the first one here. The first one is it's called phagocytosis. Phagocytosis. Okay, so phagocytosis, it means cell eating. Cell eating. Okay, the cell is literally bringing in either a lot of something or something really big. All right, so I'll give you an example. This is a white blood cell, WBC, white blood cell. Okay, and white blood cells help defend the body against invaders like bacteria. So let's say we have a bacterium here. Bacteria. And what's going to happen is the white cell will eat the bacterium. Okay, it literally will turn into like Pac-Man, that game where the yellow guy eats the dot. It's exactly like that. The bacterium will be surrounded by the cell, and before you know it, it's going to be inside the cell. Okay. If you have been reviewing your notes, do you remember what organelle will help digest this bacteria? Which organelle is going to come and help digest things that you bring into the cell? Anybody?
I'm gonna take a wild guess and say enzyme.
Right, the thing I'm thinking of has a lot of enzyme in it. Okay, but what is the name of this circle thing that's gonna have these enzymes? Not the Golgi body. Good try, Cheyenne. Yes, Trisha, it's the lysosome. Okay, so the lysosome is gonna come and then it's gonna help break down the bacteria. Okay, so everything is kind of connected. For better or for worse, guys. Okay. Now, I'm going to tell you one extra thing. This is just kind of like cool fact. You actually will learn this again next semester in the immune system. It's going to be way worse next semester, guys, in terms of content and information overload. So, take this as an opportunity to train yourself in remembering a lot of things. And so, the white cell, after they break down the bacteria, they actually will put the bacteria on its surface, like bits and pieces of it. Okay. This is for real. I'm not making this up. And they'll put it on the surface and they will show it to other white cells. They're really, proud of their achievement. They're like, hey, I just killed this bacterium. Look, like it looks like this. It's like this painting, okay? So you should go look for it and kill them because they don't belong to the body. Okay. Of course, that's like an oversimplification, but that's basically what they do to inform other white cells so they can hunt down the bacteria that is infecting the body. Okay, so that's phagocytosis, cell eating. Now, if there is cell eating, you can probably guess there will also be cell drinking, and that is called pinocytosis. These words are very, very long because a lot of them are based on, like there's like a Latin root to it in a lot of cases. And this literally means cell drinking. Okay, cell drinking. Cytosis is cell, phago is eat, pino is drink. This is cell drinking. Okay, so in cell drinking, instead of bringing in like some kind of solid like a bacterium, this is just bringing in fluid for the most part. So this is an example. The human egg can absorb fluid from its surrounding to obtain energy or nutrients rather. Oh my goodness, what happened? Ooh, let's see if I can get all those things back. Okay, from surrounding to obtain nutrients. Oh boy, let me see. Okay, the G is not disappearing. I don't know why I keep on trying to erase it. It just won't go away. Okay, just know that I'm writing nutrients. Okay, nutrients. Okay, next, last one. I don't know. Like, does this show up on yours? It's like not going away. Nutrients. Okay, there. I don't know why I cannot erase the G. Oh, there it goes. Anyway, the last type of transport that exists that you have to know is receptor. Receptor mediated endocytosis. Receptor mediated endocytosis. I'll just draw it out to explain to you. It's much easier that way than writing it out. So this is the cell membrane, as you should know by now. And on the cell membrane, there are little receptor proteins. Okay, we learned about receptor proteins last class, but these are like a little bit different. These are protein receptor. And what happens is that there are different things in the cell's surrounding. Okay, so maybe there are some like purple balls. Okay, maybe there are some, let's use a different triangle. Maybe there are some like, you know, pink triangles or something like that. Okay, and then let's just add some blue squares just for completion. Blue squares. Okay. Now, with receptor-mediated endocytosis, only molecules that match the shape of the receptor can enter the cell. Okay? So over here, only the purple balls can be brought in, not the squares, not the triangle, because they won't fit into the receptor, right? Okay? It's like, that toy that kids play when they're like, I don't know, 6 months old or something, I forgot. But oh, sorry. I don't know what's going on with the program. It's like jumping all over the place. It's like this toy, right? Where there are different shapes, and then you gotta put the shape in. Okay? Each slot is like a receptor, and only the shape that fits, that match the shape, will be able to go into the box. Otherwise, it can't come in. And that's basically what receptor-mediated endocytosis is. Right? The advantage of doing receptor-mediated endocytosis, as opposed to the other two kinds, is that there are more control as what you can bring into the cell. There are more regulation, so it's not happening by random, right? That is all for the lecture 4 notes. Are you guys okay? Any questions at all about this? I know it's a long note, but I encourage you to read through the PowerPoint slides. Okay, so you can see the PowerPoint slides are here. It's the same thing that I try to explain to you. They just have nicer diagram is all, right? Again, like I think instead of me reading the slides to you, it's there is a higher chance you'll remember it if you draw it out with me. Okay. Now there are a lot of I'm going to talk about the beetroot experiment in a bit. There are a lot of concept track questions. I'll post the answer for you to check on your own. There is one particular one I want to go through with you though. Okay. Which is like this one.
I have a question, sorry.
Yes, go ahead,
Is
Micah.
there like any animations of what we just went over, like in
Yeah,
lecture
yeah, yeah.
three?
So like a lot of people don't know this, even though I have been telling them about it. If you go to the content, and if you go to lecture, this is lecture 4, right? So each lecture folder has study resource in them, and there are a bunch of video resources that I've collected from the internet, right? So that's what you can go see. Okay, it's loading right now. And you know, there are different videos. Like there are animation for tonicity, there is animation for potassium pump, diffusion, and so on and so forth. Okay, so if you need to watch it, please go ahead and do that. And sometimes the links are broken, maybe. If that's the case, just let me know. I guess sometimes people take them offline. Uh, but yeah, I just noticed this one is missing. But the rest should be good. Is that okay?
Yep, thanks.
All right. Now I do want to go through this question with you guys, okay? Because this is like knowing the answer to this is going to help you do a question in the lab. Um, so let's take a look at this first, okay? I should I even do a poll? I don't know. You guys have not been participating too much lately, but I'll do a poll, see how many of you answer. Now, first of all, before you answer, like one person answered already, but and there are more people answering. That's great. But I just want to quickly go back here to show you. That's what we did last time, right? With the isotonic, hypertonic, and the hypotonic. Okay. So in a hypertonic solution, the cell will shrink. If it's in a hypotonic solution, the cell will burst. If it's an animal cell that we're talking about. So here, after freezing and thawing some animal cells, what would happen if they are placed in a hypotonic solution? Okay, so I see, not bad. Half of the class answered. Um, so like a lot of people chose this one, okay, because like I said, right? You see the word hypotonic and you look at the notes and you're like, oh, it's gonna swell up and burst. Okay. But the tricky part about this question is the fact that we are freezing the cell first and then thawing it. Okay. Now, I don't know if you know this, but if you freeze cells, if you freeze water, what happens to the water? Yes, I mean, like, yes, it turns into ice, but like, what happens to the volume of the water when you freeze it? Does anybody know? Like, if you fill up a water bottle and you freeze it, and you take it out after, what do you notice about the water bottle? Yes, it expands, right? So water actually will increase in volume when you freeze them. And most of the cell is made up of water, the cytoplasm, right? It's made of water. So when you freeze the animal cell, listen carefully, okay? This is very important for you to understand and maybe write down. If you're watching the recordings later to find this section, it will be at the 40, what is it, like the 50-minute mark, okay? Because you will need this answer for your lab, right? So when you freeze the animal cell or plant cell, it doesn't matter whatever you're freezing, the water will expand inside the cell, and the expansion, along with the formation of ice crystals, will actually pierce the cell membrane. Okay, when the cell, when the ice, when the water freezes in the ice and it expands, and those ice crystal that is formed, they act as like little pins and sharp edges that will cut the cell membrane, basically. So after you thaw the cell is already dead, and there are a bunch of holes in the membrane. So it actually couldn't care less whether you put it in hypotonic, hypertonic, or isotonic, because the cell membrane is destroyed, basically. So there are no regulation of what comes in and what goes out, right? So there's no change in the size. It's basically full of holes, and things just goes in and out, at equal rate. Does that make sense? Now, usually people have a question about this and fertility. Um, does anybody have a question about fertility related to this concept at all? I just want to see if anybody can make the connection here.
Is it when they freeze the eggs?
Yes, that's the question I've been waiting for. Do you want to ask the question, or do you have no question? I'm
No, I don't
forcing
have
you
the
to ask it.
question. I
No.
just guessed.
Yeah, that's great. Okay, like so like people sometimes usually you hear this for celebrities, right? Because they're the ones who can afford it. They might freeze their egg. People can freeze the sperm in sperm bank as well. So that they can have kids later on in life, right? Because one of the things that happens to a woman's egg is that they tend to deteriorate in quality over time, and women who have kids later on in life has a high chance of having chromosomal mutations in the child. So people who can afford it and they chose to do this, they can freeze the eggs while they're still young, and then they could fertilize the eggs later on in life, right? But here's the question that sometimes people would ask me. How come the eggs don't die, right? Like here in this question, I'm telling you, if you freeze it, water expands, forms ice crystal, pierce the cell, and it's going to be dead, right? So how come it's okay to freeze the egg and the sperm? And the answer is very simple. They actually add a bit of antifreeze in the solution. Okay, a lot of animals actually produce natural antifreeze. I'm not talking about the antifreeze in your windshield fluid, right? Because those, as you can see on the bottle, it says toxic, right? Poison, don't drink, right? So we are talking about like biologically safe antifreeze, like a lot of like things like amphibians, frogs, they make their own antifreeze. So in the winter, when they bury themselves like deep in the snow, they don't freeze to death, right? Because of these antifreeze. So the antifreeze basically blocks the formation of ice crystal while maintaining a cold temperature to store the eggs, and that's why it's okay. And now we know. Okay, so that's a little bit of connection for you. Okay, so I will post the answer for all the other concept check questions for lecture 3 and 4 actually, so you can study and prepare for quiz number 2. Quiz number 2 is not going to happen anytime soon, okay? It's not next week. I think it's the week after. I will let you know, okay? So no class for next week at all. You just use the time to study for the quiz as well as to do the lab, okay? To do the lab. And I just want to spend a little bit of time telling you about the lab, okay? I haven't posted an announcement for the lab yet, but I will later on, excuse me, after class. There's going to be a link where you can click into it. But it's going to be done using the quiz platform, okay? There's no lockdown browser for the lab, even though it's done through the quiz platform. It's just easier for me to mark. That's why I do it like that. And here I can preview for you over here. So once you click into it, it's going to be available to you from between now and October 3rd. Okay, so you can do it anytime between now and October 3rd. Okay, you just have to make sure you hand it in before the end of next Thursday. Okay, and then it's gonna be fine. And so there is a time limit of 3 hours. Okay, so you can't work on it a little bit every day. Okay, you have to find a 3 hour block where no one's gonna disturb you. Okay, like no interruption, and then just sit through it. 3 hours is way more than enough time, okay? Most people are able to finish this 1 hour, hour and a half tops, okay? But I know like some people, maybe you need more time to type, maybe you need more time to, review your answers, whatever. So I'm giving you 3 hours, but that's like double the time that's actually needed to do this lab, okay? So it shouldn't be a problem. Um, so when you're ready, you will click start the quiz. As soon as you start the quiz, the timer is going to start clicking right on the top right. Okay. There are 20 questions in total. Okay, so pace yourself and do them. Uh, and this is what you need to do. Okay, there are videos that you have to watch. Okay, some videos are longer than others. Like the first one you have to watch is 8 minutes. It's just me, okay, in the video talking to you about things. And the questions that you have to answer, the answers are in the video. Okay, the answers are in the video. If you watch the video, you will know the answers. Okay, I do not want you to search these stuff up on the internet. Okay, not because of plagiarism or whatever. Like obviously that's important too, but because the questions are specific to what I tell you. Like if you just try to search this up online, you will get either wrong answers or you will get things that are irrelevant to what we are talking about. Okay, so a lot of times people don't bother watching the 8 minute video and then they find some things online and they paste the answer there. And I would know because the answer is really out of place. Okay, I've been doing this for over 15 years, so I've seen a lot of weird answers from the internet about this lab. Okay, so do yourself a favor, just watch the video and then, I mean, look at the question first and then watch the video so you know what to look out for, right? Does that make sense? Right? So like, what is the purpose of this experiment? After watching the video, you will say the purpose of this experiment is to, and then you just blah, 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 right? Okay. Now, it's only 1 point, so please don't write me an essay, okay? I don't need an essay. Just answer the question and then move on, right? I always tell students to consider the 3 C's, okay? Be correct in your answer, be concise, don't ramble on like I'm doing right now, and be clear, okay? So that's what I'm looking for. And that's it, okay? Next page. And then you have to watch 3 videos. This is like me doing the Beetroot Lab. You would not know how difficult it was to film myself cutting things with only 2 hands, but I did. And basically, instead of you doing the lab, because it's virtual, right? So you watch me do the lab, and then you will answer questions related to the procedures, related to the setup and everything. Okay, so for example, what is the independent variable in this experiment? Now, if you forgot what an independent variable is, that's okay. I've also created help video for some of the harder questions. Okay, so you can watch it again. Okay, you can see me explain to you what it is, and then you can answer the question. Okay, if you know the answer, obviously you don't have to watch these things. Okay, these are called help videos, right? But the experiment ones you have to do. Okay, so there are more help. It would say help for this question. Okay, those are optional videos. So you answer all these things, answer, right? Pretty self-explanatory. The only thing that is going to take you some time is perhaps when we do data analysis, okay? There is a question 14, where you have to create a line graph, okay? I was told, did you guys learn how to do line graph yet? My other class told me they did. Did your computer teacher, Damon is your computer teacher, right? I think, did he teach you how to do a line graph yet?
No, we've only done pie graphs and just the regular bar graph.
Regular bar graph. Okay, so whatever you do not give me a bar graph, do not give me a pie graph, okay? Because this specifically asks for a line graph, and that's okay. If you don't know how to do it, there is a video that shows you how to do it, okay? Again, it's me, it's my video, okay? I explain it to you, okay? Don't go online and try to search up how to do a line graph. Just watch this one. It's only 4 minutes. You just follow the steps and you can eat. Yeah, this one is Google Sheet. If you know how to use Excel, fantastic. But Excel, some people don't have it, right? Google Sheets is free, and all of you can access it, no problem. Okay. So once you have it, you can upload your graph for me. Okay, you can take a screenshot. I don't want you to graph by hand. Okay, like in the past, I was like really messy and not professional looking. Right? Just follow the video, okay? And make sure you label the graphs. This thing is 4 marks, okay? 4 marks. You need to label the graphs, and you need to have the title and everything, okay? Is it true that you guys have to post screenshots and pictures for your math tests?
Yes, we do.
Okay, so
Yeah.
all of you would know how to upload things, right? Okay, so it's no different here. You can click on the little, insert link button. There's add file. Okay. Yeah. So just upload your graph, and it'll be okay. And then these questions are related to your graph, right? So obviously you have to have the correct graph in order to answer them. There are some, thinking questions and questions for analyzing your data and then making a conclusion. And that's it. It's just 20 questions, okay? Don't rush. 3 hours, like I said, is a lot of time, okay? Just take your time and do it, and then you will do good, okay? It's 10% of your final grade. Excellent way to move your grade up. Yeah, this is due before, like on Thursday, right? But you can do it anytime between now and Thursday. No class next week. Any questions about the lab or anything in general? Great. Okay. So I will be seeing you guys in 2 weeks. Now, if you need extra help for anything, since we don't have class next week, you can book an appointment to meet with me online. Okay. Like, if you need me to explain certain things again with you, that's fine. Just send me email and then we will set up a time that works for both of us, probably during our regular class time, right? Next week. And I'll be happy to help you. Okay. That's it. Thank you for coming. See
Thank
you
you.
next time. Bye bye.
Thank
Bye.
you.